Nutrients from non-plant sources. There are two primary ways that insect herbivores obtain supplemental nutrients from sources other than their host plant, namely by feeding at higher trophic levels and/or from symbionts, microorganisms living in intimate association with their insect host. An extensive literature documents cases of herbivorous insects, occasionally or frequently feeding at higher trophic levels, where nitrogen is more concentrated in the diet. McNeil and Southwood 1978, Polis 1981, Whitman et al. 1994, Douglas 1998, Agrawal et al. 1999a, Colin Gershon 2002, Dino and Fagan 2003, Figure 4.3. These instances include cannibalism, intraguild predation, scavenging on carcasses and feeding on other nitrogen-rich food sources such as dung. A vast array of herbivores exhibit these behaviors, including Orthoptera, Hemiptera, Thysanoptera, Lepidoptera, Coleoptera, Diptera and Hymenoptera. Detritivores such as Trichoptera engage in cannibalism and predation as well. Wissinger et al. 1996. By supplementing their diets with nitrogen from other sources, both herbivores and detritivores can increase their growth and fecundity remarkably. Anderson and McFadden 1976, McNeil and Southwood 1978, Colin Gershon 2002. Upon hatching, many first instar caterpillars, e.g., Aschamonas, seek nitrogen-rich meals by regularly consuming their own egg chorion or the egg soft nearby conspecifics, Bajos belinda and Zucalito 2001, whereas other taxa, cockroaches and lepidopterans, often consume their exuvia and partially recover lost nitrogen, Mira 2000. Factors that further motivate herbivores to seek nutrients from other sources include food depletion, reductions in plant quality, high population density and physiological state, such as when females are in the process of maturing eggs, Simpson et al. 2006. If plant quality is poor and nitrogen is not available from other sources, females may resorb eggs or embryos, thereby enhancing their own survival. Ogushi 1996, Omak and Leather. 2002. Numerous orders of insects have member species that harbor a variety of symbiotic microorganisms such as bacteria, fungi and protozoans. Bortses and Miller 2006. Symbiotic mutualists are completely dependent on their hosts, but in turn provide them with nutrients, e.g., amino acids, sterols and vitamins. Chemicals that are either rare, absent altogether, or tied up in non-digestible forms in plant diets. Liados et al. 1995, Bowman et al. 1997, Douglas 1998, Bortses and Miller 2006. Symbionts are overrepresented in groups of insects that feed on nutritionally poor food or imbalanced diets, suggesting that housing symbionts is an adaptation to meet nutrient demands. For example, symbionts occur in phloem and xylem feeders, Hemiptera, aphids, psyllids, whiteflies, scale insects, planthoppers, leafhoppers and cicadas, some folivores, Hymenoptera, leafcutter ants, wood feeders in the Coleoptera bark, ambrosia beetles and some scarab sandweevils, and Isoptera termites, and of faunivores, Blatodia, wood roaches. Bowman et al. 1997, Gullin and Cranston 2005, Bortses and Miller 2006. Notably, symbionts are not known to occur in predatory insects that feed on more nitrogen-rich food. Microbial symbionts can occur external to the insect's body, ectosymbiotic fungus grown by leafcutter ants, or within the host, endosymbiotic gut protozoans and termites, and can be either extracellular bacteria and protozoans in the guts of termites and cockroaches, or intracellular bacteria in the specialized mycetome cells of aphids. See Chapter 6 on mutualism. The role of symbionts in host nutrition has been examined by ridding hosts of symbionts with antibiotics, heat shock treatments or otherwise sterilizing them and then comparing the performance of so-called aposymbiotic hosts with symbiotic control groups. Chen et al. 1981, Campbell 1989, Douglas 1996, 1998. When symbionts are deactivated, hosts grow slower, molt to smaller adults or exhibit reduced fecundity, a result that has been shown for aphids, planthoppers, bark beetles and termites. Chen et al. 1981, Fox et al. 1992, Yoshimura et al. 1993, Liados et al. 1995. Moreover, some herbivores such as leafcutter ants, e.g., Atta culture fungal symbionts in their subterraneanists and bark beetles, e.g., Ipes, Dendroctonus, and ambrosia beetles, e.g., Platypus vector theme among host trees during colonization. Beaver 1989, Charette et al. 1989, Fox et al. 1992. Depending on species, these herbivores feed on the ectosymbiotic fungus either exclusively Atta, or in part Ipes, thereby providing essential nutrients for growth and population increase that are available in very limited amounts in leaf tissue or wood. Termites as a group have evolved a variety of mutualisms with different microorganisms that aid in wood digestion, nitrogen acquisition and ultimately balancing their own C/N content when feeding on very carbon-rich food. Bresnak 1982, Higashi et al. 1992. For the most part, gut microorganisms such as protozoans, bacteria and fungi are required for cellulose digestion. Bresnak 1982, Martin 1991. Moreover, several lineages of termites enhance nitrogen intake by harboring bacterial-gut symbionts that either fix atmospheric nitrogen or synthesize it. Higashi et al. 1992, Moria et al. 1999. Some termites also support methanogenic bacteria in their guts and are thus able to eliminate excess carbon by methane production. Higashi et al. 1992, Brahman et al. 1992. Overall, the symbiosis that termites have evolved with microbes are essential for maintaining their own C/N stoichiometry. Symbionts at large provide herbivores with essential nutrients, particularly nitrogen that is so critical for growth and reproduction. Notably, symbiotic relationships with microorganisms appear to have provided the opportunity for insects to exploit and diversify in nutrient-poor resources that are otherwise very difficult to exploit. See Chapter 6 on mutualism. Evolutionary shift in body nutrient composition. Another way that herbivores have partially reduced their chronic demand for nitrogen is to evolve a lower dependence on nitrogen for the construction of body constituents, and thus reduce the stoichiometric mismatch with their plant diet. Fagan et al. 2002. At the level of protein subunits, selection may favor amino acids with lower nitrogen contents. Recent research on the elemental composition of bacterial anti-yeast proteins has shown that shifts in protein composition can evolve in response to elemental shortages. Badwan Cornu et al. 2001. At the tissue level, selection might, for example, reduce the ratio of protein to chitin in insect cuticle that typically ranges from 1 to 4. 1 Chapman 1998. At the whole body level, selection might alter the relative allocation to muscle, cuticle, fat body and other tissues, all of which differ in nitrogen content. Fagan et al. 2002. That herbivorous bugs and beetles have thinner cuticles than their predaceous counterparts is in line with this argument. Rees 1986. Selection at all of thessa levels of organization may explain why insect herbivores have lower nitrogen content, 9.6%, on average than predators, 11.0%. Fagan et al. 2002, Matsumura et al. 2004. Similarly, selective pressures associated with fundamental limitations in atrogen may have contributed to reductions in body nitrogen content over evolutionary time. Consistent with this hypothesis is the observation that herbivores in the more derived orders of insects, Lepidoptera and Diptera, contain 15-25% less nitrogen than do those in more basal lineages, Orthoptera and Hemiptera. Fagan et al. 2002. In this context, half of the body mass of rigid grasshoppers is proteinaceous cuticle, which is far more than that for flexible caterpillars. Bernays 1986. Thus, nitrogen conservation may have played a role in the evolution of holometabolic complete metamorphosis, with the development of a thin cuticle in the larval stage of advanced insect orders. Bernays 1991. 4.3.3 Mechanical and structural barriers to herbivore attack. Besides being nutritionally inadequate, plants possess a variety of mechanical features and structures that pose physical-chemical barriers to herbivore attack. Included in the repertoire of mechanical and structural defenses are general tissue toughness and hardness that deter or prevent feeding. Lucas et al. 2000. Trichomes plant hairs that can deny or reduce herbivore access to feeding and oviposition sites. Myers and Basili 1991. Andres and Connor 2003. And surface waxes that can make it difficult for herbivores to colonize and maintain their foothold on plant surfaces. Juniper and Southwood 1986. Eigenbrode and Espelli 1995. Historically, ecologists have referred to leaf toughness as a general mechanical defense against insect herbivores, but usually the structural elements conferring tissue resistance are not explored in detail, e.g., Feeney 1970, Coley 1983, Raup 1985, but see Peters 2002. In general, young expanding leaves are less tough than are mature leaves, and leaf thickness and the amount of cellulose and lignin, the structural components of plants, have been implicated in tissue toughness. Peters 2002. Recently, generic tissue toughness has been partitioned into two components, namely hardness and toughness, both of which can have adverse effects on herbivores. Chung et al. 1992, Lucas et al. 2000. Hardness deters the initial cracking, splitting of a plant tissue when a herbivore begins to feed, whereas toughness results in resistance to crack growth. Thus, a seed coat may be very hard, resist cracking, but be very brittle and therefore not be very tough. The primary source of tissue toughness is composite cell wall consisting of cellulose microfibrils sit in a hemicellulose or lignin matrix, and is roughly proportional to the fraction of plant tissue volume occupied by cell walls. High toughness in plant cells results not from the cell walls themselves, but rather from their plastic ability to collapse. Hardness in plant tissues can be achieved by dense cell walls as in some seed coats or by amorphous silicane leaves, spines or surface structures such as stiff trichomes. Such defenses deter herbivores from contacting plants, but are also responsible for significant abrasion and mandibular wear. Onsfeeding has begun. Lucas et al. 2000. For mandibulate herbivores such as lepidopterans and beetles, feeding on tough leaves results in reduced consumption and delayed growth, which in some cases can be attributed to increased mandibular wear. Raup 1985, Stevenson et al. 1993, Figure 4.6. Figure 4.6. Left mandible of an adult willow leaf beetle Plagiodora versicolora that has fed for 1 month on A. Tender terminal leaves or B. Tough mature leaves. The incisor of the mandible is completely worn down when beetles feed on tough leaves. From Raup 1985. Reprinted with permission from Blackwell Publishing. Similarly, leaf shredding caddisflies can show decreased consumption rates on tough litter types. Motomori et al. 2001. In several cases, mandibular wear and impaired growth in stem-boring caterpillars have been linked to the high silica content of their grass host. Potic et al. 1971. Hanifa et al. 1974. Thus implicating tissue hardness as the underlying mechanism. Even more convincing are studies in which the silica content of the host plant was experimentally increased with adverse effects on the growth and digestion efficiency of grasshoppers, lepidopterans and dipterustem borers. More 1984. Massey et al. 2006. Notably, sap-feeding herbivores, aphids, in this study were not affected by elevated silica. Sap feeders may more easily penetrate the cellular spaces between silica-containing cells, and as a consequence are not physically excluded from feeding in the vascular tissue. This might explain why herbivore communities on silica-rich grasses are filtered, largely devoid of money-chewing herbivores, e.g., free-living lepidopterans and beetles, but not grasshoppers, and are dominated by sap feeders such as leafhoppers and planthoppers. Cook and Dino 1994. Recent evidence, however, suggests that sap feeders to incremandibular stylet wear while feeding in plant tissues. Royberg et al. 2005. But mandibular wear has yet to be compared between chewing and sap feeding herbivores fed the same diet. Notably, the structural traits of leaves, e.g., blade and cuticle thickness, vein linification and thickened hypodermis, have also been shown to influence the guild structure of arboreal insect assemblages, Peters 2002. For example, the density of leaf-chewing herbivores was negatively correlated with a thickened hypodermis and the area of the leafweed lignified. In fact, in this extensive study, the functional composition of the herbivore assemblage was better correlated with structural leaf traits than with leaf constituents such as nitrogen and water content. Overall, tissue toughness, hardness and surrogate variables have significant effects on individual performance, population density and community structure. Trichomes occur in a diversity of forms, sizes and densities and in part serve to protect plants from herbivore attack, although there is substantial variation in their effectiveness. Myers and Basile 1991, Peter and Schnauer 2001, Heron L 2002, Andres and Connor 2003. In addition to their anti-herbivore role, trichomes also insulate leaves from solar radiation, deter evaporation, facilitate water and nutrient absorption, or function in salt excretion. Gutchick 1999. Overall, trichomes affect insect herbivores by influencing oviposition, altering herbivore movement, reducing growth and fecundity, and by influencing interactions with natural enemies. Haddad and Hicks 2000, Andres and Connor 2003, Kennedy 2003. Simply, trichomes can be divided into two general types, namely non-glandular and glandular. Levin 1973, Hare 2005. Multiple trichome types can occur on the same individual plant. The same trichome type can vary in density and size among individuals and populations, and some plant species are dimorphic for trichome type with glandular and non-glandular trichomes occurring in different individuals. Heron L 2002, Kennedy 2003. Non-glandular trichomes physically interfere with feeding and colonization, especially for small insects such as first instar lime aphids, Eucliteris tillii, that die because they are denied access to the leaf surface where they reach the phloem. Dixon 1998. When the dense bed of stellate trichomes is removed, the young aphids feed and flourish. Similarly, shaving the dendroid trichomes from mullein verbascum leaves promotes colonization by the aphid Aphis verbascae, Keenleyside 1989. Other small sap-feeding herbivores such as whiteflies and leafhoppers also fail to colonize or successfully grow on cultivars of crop plants rendered resistant by dense beds of leaf trichomes. Butler et al. 1991. Gortson and Small 1993. Larger herbivores too can be very adversely affected by non-glandular trichomes. For instance, larvae of Heliconius melpomene that consume a number of passion vine species Passiflora are excluded. For instance, larvae of Heliconius melpomene that consume a number of passion vine species Passiflora are excluded from the widespread Passiflora adenopoda. Gilbert 1971. The leaves and tendrils of this vine are covered with hooked trichomes. As larvae attempt to feed, their prolegs catch on the trichomes and tear. Hemolymph exudes and the larvae quickly desiccate. Andy, Figure 4.7a and b. Similarly, the growth and survival of the swallowtail butterfly, Papilio troilus, and several noctuid moth caterpillars are negatively Similarly, the growth and survival of the swallowtail butterfly, Papilio troilus, and several noctuid moth caterpillars are negatively affected by leaf pubescence, and in some cases reduced performance and survival occur because trichomes are of lower nutritional quality than other leaf tissues. Lambert et al. 1992, Haddad and Hicks 2000, Andres and Connor 2003. It should be emphasized that not all herbivores are negatively impacted by trichomes, and in fact some herbivores prefer plants with dense trichomes for oviposition, and if-adapted perform better on such plants. The exudates from glandular trichomes can deter, Meyer or poison insect herbivores, Ranger and Howard 2001, Andres and Connor 2003, Kennedy 2003. Small herbivores such as aphids and leafhoppers become entrapped and die in the sticky exudates produced by glandular trichomes on their host plants, Dixon 1998, Kennedy 2003, Figure 4.7c, D and E. However, the exudates of glandular trichomes also contain toxins, which in the case of tomato confer resistance to a variety of herbivores including aphids, whiteflies, lepidopterans and dipteran leaf miners. Although the physical deterrency of non-glandular trichomes is clear, it is often difficult to isolate the effects of mechanical defense, allochemistry and nutrition when glandular trichomes are involved because the battery of plant defenses is so intimately intertwined. Scaling up to herbivore communities, an extensive study of the insect guilds on Manzanita Arctostaphylos species showed that leaf pubescence has both community-wide and guild-specific effects on folivorous insects because of its selective effects on free living, but not concealed, feeding guilds. Andres and Connor 2003. Feeding by herbivores can also induce the production of trichomes on the new growth of their host plants, with adverse consequences not only for the inducer, but also for the community of other herbivores feeding on the plant. Bauer et al. 1991, Agrawal 1998, 1999, 2000a, Trawan Dawson 2002. Induced defenses at large will be dealt with in a forthcoming section of this chapter. Leaves of some plant species also bear other surface structures that deter herbivore attack. For example, protrusions on the stipules and meristems of Passiflora bear an amazing resemblance to the eggs of Heliconius butterflies, the primary herbivores on these plants. Benson et al. 1975, Williams and Gilbert 1981, Figure 4.8. So-called egg mimics significantly reduce the number of potential oviposition sites because adult butterflies avoid placing eggs in their presence. Selection has apparently favored oviposition site scrutiny in these visually oriented butterflies because upon hatching larvae are very cannibalistic and consume other nearby eggs. In contrast to plants with trichome-laden surfaces, many plants have leaves rendered slippery by the surface waxes that provide protection from desiccation and pathogen invasion. Eigenbrode and Espelli 1995, Eigenbrode et al. 2000, Rutledge et al. 2003. Such waxes often pose problems of attachment for many herbivores. For example, glossy varieties of cabbage, Brassica oleracea, deter foraging and feeding by larvae of the diamondback moth Plutelixilostella, caterpillars of the imported cabbage butterfly Pieris rapi, and cabbage aphids Brevicorine brassicae, whereas other herbivores such as flea beetles Philotreta cruciferae are able to maintain a foothold. In the cabbage system, complex interactions among structure of surface waxes and the tarsal morphology of insects combine to influence attachment. Eigenbrod and Jeter 2002. Similar effects have been documented in natural systems, whereby young leaves of some eucalyptus species possess a waxy bloom that precludes attachment and feeding by beetles. Edwards 1982. Overall, the distribution and abundance of leaf. Figure 4.7a Caterpillar of Heliconius melpomene caught on the hooked trichome. Figure 4.7a Caterpillar of Heliconius melpomene caught on the hooked trichomes of Passiflora adenopoda. B. Close-up of trichomes hooked into the caterpillar's proleg. From Gilbert 1971. C. A glandular trichome of an alfalfa clone resistant to potato leafhopper, Emposca fabi A1000, which has released exudates after damage. D. A nymph of the potato leafhopper entrapped in the glandular exudates. From Ranger and Hauer 2001. E. Adult of the aphid Rhopalosiphem matus entrapped in the latex of its lettuce host plant, Lactuca sativa. From Dussord 1995, Dr. David Dussord. A. and B. From Gilbert, L.E. 1971. Butterfly plant coevolution. Has Passiflora adenopoda won the selectional race with heliconine butterflies? Science 172, 585-586. C. and D. Reprinted with permission from Blackwell Publishing. See color plate section. Feeding beetles on Eucalyptus spp. Is related to the waxy bloom on the trees and the attachment abilities of the individual beetle species. Edwards and Wanjera 1991. Although waxy leaf surfaces and dense trichomes often deter some natural enemies, not all predators and parasitoids are adversely affected. Bottrell et al. 1998. Eigenbrod and Jeter 2002, Kennedy 2003, Eigenbrod 2004. For example, some natural enemies are better able to negotiate agricultural crop varieties with reduced surface waxes on their leaves, consume more herbivores and thus exacerbate the direct adverse effect of slippery leaves on herbivores. Likewise, fire ants Solenopsis invicta are not Dipteroides soybean plants with dense trichomes and in fact suppress Lepidopteran caterpillars better on pubescent varieties than on glabrous ones. Stiersky et al. 2006. Thus, to assess the general effect of plant surfaces with abundance of insect herbivores, one must take into account the direct effects of surface structure on herbivore attachment and the indirect effects of altered foraging by natural enemies. 4.3.4 Herbivore counteradaptations to mechanical plant defenses. Insect herbivores have evolved a number of morphologies and behaviors that allow them to cope in part with the mechanical defenses of plants. Concerning leaf hardness, insights can be gained by comparing the morphology of herbivores that feed on silica-rich grasses with that of forb feeders. For instance, the relative head and associated mandibular mass of grass-feeding grasshoppers and Lepidopterans is larger than that for related forb feeders. Bernays 1986, and egg size in the Satyridae and Hesperiidae Lepidoptera is positively related to the leaf toughness of their host grass. Fukuda et al. 1984, Nakasuji 1987. Larvae hatching from large egg shave large heads and mandibles which retain their cutting and masticating capability until worn mandibles are renewed at the next molt. Thus, large head and mandible size has apparently allowed some herbivores to exploit grasses, but the price these specieshave paid for this dietary habit is reduced fecundity. Also, several species of grasshoppers, e.g., the grass-feeding Scherthippus brunnius and Figure 4.8 Protrusions on the stipples of Passiflora cyanea bear a remarkable resemblance to the eggs of Heliconius butterflies, the primary herbivores on this group of plants. Adult butterflies avoid placing their eggs in the presence of so-called egg mimics, significantly limiting the number of potential oviposition sites and ultimately reducing herbivora. Extrafloral nectaries B are sugar-secreting glands that provide carbohydrate resources to ants and other predators that further reduce the density of insect herbivores on P. cyanea. Original from Gilbert and Rabin, 1975, adapted from Strong et al. 1984a. Lepidopterans, e.g., the gypsy moth Lymantria dispar, can undergo extra molts during their immature development. Supplemental molting allows individuals to replace worn mandibles, and may allow for the exploitation of hard leaf tissues. Moreover, the mandibles of grass-feeding grasshoppers have chisel-edged incisors, and a well-developed molar region for grinding, whereas forb feeders do not. Bernays 1991. Herbivores also have evolved tarsal morphologies for negotiating trichome-bearing leaf surfaces. Some aphids and myrid bugs, for example the specialist oak-feeding aphid, Mysicalis scriberi, have tarsal claws modified for grasping trichomes and moving through the densely pubescent leaves. Kennedy 1986, Southwood 1986. Several aphids exploiting host plants with glandular exudates have short tarsi or no tarsi whatsoever, an adaptation which apparently allows them to tiptoe through the trichomes. Moran 1986, Bernays 1991. Alternatively, other aphids have solved the problem of accessing the leaf surface by evolving a longer proboscis, which allows them to feed through the dense bed of trichomes. Dixon 1998. Larvae of the neotropical butterfly, Mechanitis isthmia, feed on Solanum sp. whose leaves are covered with dense beds of trichomes. Rathke and Poole 1975, Young and Moffat 1979. Eggs are deposited in masses and upon hatching larvae aggregate on the lower surface of leaves where they collectively spin a silken scaffolding over the top of the trichome bed. Subsequently, larvae roam the top of the silken mat where they clip trichomes and safely consume leaf tissue. Both mandibulate and sap-feeding herbivores have evolved tarsal modifications that allow attachment on waxy leaf surfaces. Bernays 1991. For instance, some chrysomelid beetles, like gecko lizards, are able to hold onto glossy leaf surfaces because of the molecular adhesion provided by thousands of minute setae on their tarsal pads. Stork 1980. Likewise, certain Amposca leafhoppers produce a minute suction cup with their tarsal pads, which provides very effective attachment on smooth leaf surfaces. Lee et al. 1986. Notably, the tarsal adaptation works well in preferred glossy leaf surfaces, but is ineffective in pubescent leaves. 4.3.5 Allelochemical barriers to herbivore attack. The insecticidal properties of plants have been known for several centuries. For instance, water extracts of tobacco, Solanaceae, were used to kill sap-feeding insects in 1690. Rhodonone fabaceae was used to kill caterpillars in 1848, and Pyrethrum astraceae has been used as an insecticide since 1880, Ware 1991. Moreover, the active fractions of these botanical insecticides were all isolated prior to 1924, Matsumura 1985. Despite the long-standing knowledge of the toxic properties of plants by entomologists and natural product chemists, the role that secondary metabolites, also called secondary chemicals or allelochemicals, play in plant-insect interactions has been realized relatively recently. Bruce, 1946, Painter, 1951, Frankel, 1959, 1969, Ehrlich and Raven, 1964, and Feeney, 1968, 1970, were among the first ecologists to promote the importance of allelochemicals as defenses against insect herbivores. Since then, the literature documenting the structure, diversity, distribution, concentration, induction, metabolism and anti-herbivore properties of allelochemicals has grown enormously, e.g., Rosenthal and Berenbaum 1991, 1992, Ptolemy and Raup 1991, Harbour 1993, Carbon and Baldwin 1997, Agrawal et al. 1999b, Kessler and Baldwin 2002, Boge and Marquis 2005. Secondary metabolites are deemed secondary because they play little or no known functional role in the primary plant metabolism underlying plant growth and reproduction. Whitaker 1970. Literally thousands of secondary metabolites have been isolated from plants, and there is unequivocal and widespread evidence that these compounds serve a defensive function in plants against herbivores and pathogens. Rosenthal and Berenbaum 1991, Harbour 1993, Carbon and Baldwin 1997, Kessler and Baldwin 2002. For instance, allelochemicals have been shown to affect nearly every aspect of plant-herbivore interactions, including their preference for plants, host plant selection, and performance, growth, survival, reproduction and dispersal on particular plants or plant parts. Despite their anti-herbivore and pathogen properties, however, there is also a clear indication that secondary metabolites serve other functions, such as protection from ultraviolet radiation, e.g., flavonoids, storage compounds, e.g., nitrogen-containing alkaloids, and plant signaling compounds, e.g., terpenoids. Rhodes 1979, Chadwick and Whalen 1992, Burchard et al. 2000, Thays and Lerdow 2003. Thus, selective pressures associated with herbivorean pathogen attack are not the only forces influencing the evolution of secondary metabolites. Before exploring factors that influence the distribution and abundance of secondary metabolites in plants and the counteradaptations of insect herbivores that allow them to cope with such compounds, we need to consider the diversity of secondary metabolites and their modes of action. The tremendous diversity of allelochemicals makes it difficult to organize these compounds into a conceptual and useful framework. Most classifications of allelochemicals are oversimplified and are based on modes of action or how allelochemicals are expressed in plants. One approach has been to divide secondary metabolites into two broad functional categories based on their modes of action, namely qualitative and quantitative defensive compounds, or alternatively toxins and digestibility reducers respectively. Feeney 1975, 1976, 1992, Rhodes and Cates 1976, Rhodes 1979. Secondary metabolites have also been classified as either constitutive defenses, compounds that are continuously present in the plant and do not change following attack, or induced defenses, metabolites that are synthesized or released from pre-existing ducts or cells following attack by insect herbivores or plant pathogens. Rhodes 1985, Carbon and Baldwin 1997. Secondary metabolites. Qualitative defensive compounds such as alkaloids, pyrethrins, glucosinolates, cardenolides, cyanogenic compounds and non-protein amino acids interfere with the metabolism of insect herbivores, thus the synonym toxins. In addition to interfering with metabolism, qualitative defensive compounds are characteristically small molecules, present in low concentrations in plant tissues, less than 2% dry weight, and possess properties facilitating their rapid synthesis, storage and transport. Rhodes and Cates 1976, Rhodes 1983, Figure 4.9a. Though cheap for plants to synthesize, qualitative defenses are thought to provide limited protection against cobalt specialist herbivores that have developed detoxification or tolerance mechanisms. Notably, naturally occurring plant toxins, e.g., nicotine, often share modes of action that are similar to those of some synthetic insecticides, e.g., the neonicotinoid insecticide imidacloprid, Bloomquist 2006. For example, the pyridine alkaloid nicotine, naturally present in solanaceous plants such as tobacco, is toxic to insect herbivores because it interferes with neurotransmission. Stepan et al. 2004, Bloomquist 2006. Specifically, nicotine mimics the neurotransmitter acetylcholine by binding with acetylcholine receptors, which leads to the persistent overstimulation of cholinergic synapses, convulsions, paralysis and rapid death. Similarly, pyrethrins, derived from the flowers of Chrysanthemum cinerariifolium, Astraceae, are neurotoxins that have potent effects on nerve impulse generation. Cyanogenic compounds, e.g., HCN, that occur commonly in rosaceous plants, e.g., Prunus sp. Act on the cytochrome system. That terminal respiration is inhibited, oxygen starvation occurs at the cellular level, and rapid death ensues. Harburn 1993. Other metabolites such as the cardenolides in milkweed plants Asclepiadaceae, Noopocinaceae act as sodium-potassium ATPase inhibitors and disrupt the sodium and potassium fluxes in cells. Malcolm 1991, Agrawal 2005. NMEO Coach. GLC. Moreover, glucosinolates and their hydrolysis products in mustards and related plants Brassicaceae degrade proteins and have adverse effects on respiratory physiology. Sau et al. 2002, Whitstock et al. 2003, Hockyer and Gershens in 2006. Another well-studied group of toxins are the furanocoumarins, a photoactivated group of compounds in the parsley Apiaceae and citrus rutaceae families that derive their toxicity by cross-linking with NA strands and inhibiting transcription. Barenbaum et al. 1991, Wen et al. 2006. Quantitative defensive compounds include tannins, resins, latexes, cellulose, lignin and silica, all compounds that can act to reduce the digestibility of plant tissues. Feeney 1976, Rhodes and Cates 1976, Rhodes 1983. In fact, most insect herbivores are unable to digest cellulose and lignin at all, the major structural components of plants. Martin 1991. Typically, quantitative defenses are large molecules, e.g., tannins or large polyphenols, occur in relatively high concentrations in plant tissues, 5–20% dry weight, and are thought to be more costly to produce than toxins. Figure 4.9b. Though costly, quantitative defenses are thought to provide plants better protection against both specialized and polyphagous herbivores because they act in a dosage-dependent fashion. Although previously discussed as mechanical barriers to herbivore attack, cellulose, lignin and silica are also categorized as quantitative defenses because they reduce herbivore growth in a dosage-dependent manner, occur in high concentrations in plants, render plant tissues tough and difficult to digest, and act by inflicting mandibular wear. Feeney 1975, Rhodes and Cates 1976, Peters 2002, Massey et al. 2006. Another quantitative defense includes the tannins, a complex suite of high molecular weight phenolic compounds that occur abundantly in woody plants, both gymnosperms e.g., pines and angiosperms e.g., oaks, willows, maples, eucalypts and acacias, but do also occur in some grasses e.g., sorghum. Haslam 1989, Hemingway and Karchesi 1989, Waterman and Mole 1994. Moreover, tannins are distributed in numerous tissues such as leaves, stems, buds, seeds and even roots. Tannins occur in a diversity of forms and are often divided structurally into hydrolyzable tannins, esters of sugars with gallic acid, and condensed tannins, polymers of flavonoids. Harburn 1993, Ayres et al. 1997, Figure 4.9b. Tannins are thought to reduce the digestibility of plant tissues by precipitating plant protein from solution under certain conditions. Feeney 1970, Harburn 1993. Thus, when insect herbivores bite into tannin-rich leaves, compartmentalized tannins are brought into contact with plant proteins and digestive enzymes forming tannin-protein complexes that limit the digestion and assimilation of plant proteins. Harburn 1993, Hagerman and Robbins 1993, Zucker 1983, Ayres et al. 1997. Overall, however, there is very mixed support for the view that tannins act as a general quantitative defense against insect herbivores, with evidence both for and against. Smith et al. 1992, Hunter and Schultz 1995, Iris et al. 1997, Forkner et al. 2004. For example, specific condensed tannins differ markedly in their anti-herbivore activity and insect herbivores differ in their sensitivity to tannins. Ayres et al. 1997. In part, interspecific variation in herbivore response to tannins is likely attributable to differences in gut physiology that can ameliorate tannin effects. In part, interspecific variation in herbivore response to tannins is likely attributable to differences in gut physiology that can ameliorate tannin effects. Barenbaum 1980, Martin and Martin 1984, Apple and Maines 1995. Complicating matters further is the knowledge that some studies implicate toxicity rather than digestive inhibition as mode of action of some tannins. Zucker 1983, Steinle and Barenbaum 1985, Ayres et al. 1997. Challenging the broad distinction between quantitative and qualitative defenses. Many plants also contain proteinase inhibitors, enzymes that inhibit the action of digestive protease enzymes, trypsin and chymotrypsin, in the guts of herbivorous insects, e.g. Lepidoptera, thus making plant proteins less available and adversely affecting growth. Ryan 1990. Because proteinase inhibitors are protein-based enzymes, the distinction between primary and secondary metabolites becomes less clear, and suggests that even some primary metabolites may play a defensive role. Barenbaum 1995. Another major group of quantitative defenses are the resins and latexes, sticky plant secretions synthesized in large quantities by many plants, including gymnosperms and angiosperms. Farrell et al. 1991, Phillips and Croteau 1999, Becerra et al. 2001, Langenheim 2003. Resins are complex mixtures of volatile and non-volatile terpenoids, e.g., monoterpenes, diterpenes, triterpenes and sesquiterpenes, and orphenolic compounds that, in addition to their defensive function against insect herbivores and pathogens, also play a role in wound repair, water retention and UV protection. Latexes are emulsions, aqueous suspensions of insoluble materials that contain metabolites such as alkaloids, cardenolides, terpenes and phenolics. Typically, resins are secreted from glands e.g., creosote bush, Lariat tridentata, or trichomes e.g., hemp, cannabis sativa, one plant surface, or they can be sequestered intercellularly in resin ducts or canals such as familiar pitchy oleoresins, terpenoids, in coniferous trees, Farrell et al. 1991, Langenheim 2003, Keeling and Bowman 2006. In contrast, latexes such as milky secretions in milkweeds, Apocynaceae, and spurges, Euphorbiaceae, are contained in living cells called laticiphers. Resins can be deployed either constitutively from surface glands or, like latexes, they can be released, induced, in copious amounts when herbivores sever resin-bearing canals, a subject we will pursue in greater depth shortly. The mode of action of resins and latexes is complex and often involves both physical and toxic components. For example, many resins and latexes inhibit insect herbivores simply by entrapping them or impeding feeding by gumming up their mandibles. Thus their espoused role is a quantitative defense with dosage-dependent effects. Dussord 1995, Phillips and Croteau 1999, Langenheim 2003. In this sense, these compounds are not digestibility reducers per se, but they do physically deter feeding and may result in digestive costs as well. Additionally, resins and latexes often contain toxins or feeding and oviposition repellents, thus complicating their assignment to general plant defense categories. Dussord and Hoyle 2000, Langenheim 2003, Keeling and Bowman 2006. Another way to classify secondary metabolites is based on their expression in plants. Once synthesized, constitutive defenses are always expressed in plant tissues. There is tremendous diversity in the composition and concentration of naturally occurring constitutive defenses, ranging from mechanical defenses e.g., cellulose, silica and trichomes in some cases, to digestibility reducers e.g., resins and tannins, and toxins e.g., alkaloids and furanocoumarins. Zangrel and Rutledge 1996, Keeling and Bowman 2006. In contrast, induced defenses, also called induced resistance, are activated by herbivore feeding e.g., mechanical damage and saliva, or pathogen attack. Carbon and Baldwin 1997, Agrawal et al. 1999b, Kessler and Baldwin 2002, Tra and Dawson 2002, Tra and Bergelson 2003, Bostock 2005, Stout et al. 2006. A vast array of secondary metabolites and trichomes can be induced by herbivore feeding. Allochemicals known to be induced by herbivores include defensive proteins, proteinase inhibitors, phenolics, phenolic acids, phenolic glycosides, furanocoumarins, condensadant hydrolyzable tannins, andlinin, terpenoids, monoterpenes, diterpenes and cucurbitacins, alkaloids, nicotine, quinolizidine antropane, and indole glucosinolates. Green and Ryan 1972, Haukioha 1990, Constable 1999, Kessler and Baldwin 2002. Moreover, some compounds uncategorized as constitutive defenses are known now to be inducible, e.g., silica, further challenging the traditional classification of plant defenses. Massey al. 2006. Induction of phytochemicals by herbivores can take place in two ways, by the release of preformed chemicals, or by activated synthesis, both of which can have very negative consequences for herbivores. Carbon and Baldwin 1997, Agrawal 1999, Dino and Kaplan 2007. Unlike most activated allochemicals, the induction of preformed defenses does not result exclusively from changes in synthesis or degradation. Carbon and Baldwin 1997. Rather, the release of preformed chemicals results from the disruption of tissues where such chemicals are compartmentalized or from the mixing of locally separated substrates and enzymes during tissue damage. Examples of preformed compartmentalized allochemicals include the terpenoids stored in the resin ducts of conifers. Raffa 1991, Raffa et al. 2005, Keeling and Bowman 2006, the cardenolide containing latex borne in the canal systems of milkweeds. Dussord and Eisner 1987, and the furanocoumarins housed in oil tubes of wild parsnip plants. Barenbaum and Zangrel 1999, Zangrel 1999. When herbivores puncture such ducts, canals or oil tubes during feeding, the compartmentalized allochemicals are released, which can entrap and kill herbivores attempting to feed. A classical example involves bark beetles Coleoptera, Scolididae that sever the pressurized resin ducts Wienbring into conifers. Raffa 1991, Keeling and Bowman 2006. When punctured, oleoresins pitch out, and mire colonizing beetles. A more dramatic case involves plants in the genus Burseraceae that hold terpene-containing resins under high pressure in canals that resins can squirt out or pull up when canals are severed by marauding herbivores. Becerra 1994, 2003. Other canal systems are so sensitive to a herbivore's touch that latexes exude from laticiphers and entrap aphids Rhopalosiphamatus that simply walk on the surface of wild lettuce plants. Lactuca sativa, Dussord 1995, Figure 4.7e. Examples of preformed allochemicals that are induced by the mixing of separated substrates when plant tissue is damaged includeth blending of cyanogenic glycosides and catabolous enzymes with the production of hydrogen cyanidine. Cyanogenic plants. Palton 1990, the hydrolysis of compartmentalized glucosinolates by myrosinacetto form toxic thiocyanates in crucifers. Chu 1988, Bones and Rossiter 1996, Agrawal and Kurashige 2003, Whitstock et al. 2003, and the conversion of phenolic glycosides to more active feeding deterrents as salicin in poplars. Clausen et al. .1989. Activated allochemicals, e.g., proteinase inhibitors, alkaloids and glucosinolates, are synthesized following herbivora and result in an actual increase in the allochemical pool with adverse effects on some herbivores. Carbon and Baldwin 1997, Constable 1999, Bostock 2005, Mewis et al. 2005. Induced increases in allochemicals can occur over a range of time intervals from a few hours or days, rapid induced response, to the next season, delayed induced response. Carbon and Baldwin 1997, Agrawal et al. 1999b. Moreover, different herbivore species can induce very different levels of plant resistance with dramatically different effects on the assemblage of herbivores that feed later that season. Van Zandt and Agrawal 2004a, b. Viswanathan et al. 2005. Importantly, however, the distinction between activated and preformed induced responses is best viewed as a continuum, because some allochemicals have elements of both. For example, all taufuranocoumarins, terpenes and glucosinolates occur in preformed states. They can be also induced several-fold by herbivora, suggesting induced synthesis. Lewinson et al. 1991b, Agrawal et al. 1999b, Zangrel 1999. Induced defenses that are activated by herbivore feeding, e.g., mechanical damage and saliva or pathogen attack, are communicated through the plant via signaling pathways that ultimately result in the production of an allochemical. Felton and Eichenseer 1999, Bostock 2005, Stout et al. 2006. The best-known signaling pathway in plants involves the wound hormone jasmonic acid, which underlies increases in many putative defensive chemicals against insects. Although jasmonic acid is clearly involved in the induction of plant responses to insect herbivores, it is thought that cysteamine, a small polypeptide hormone, is the compound that actually moves through the plant following herbivora to activate the formation of jasminates and ultimately the synthesis of allochemicals. Carbon and Baldwin 1997, Stiswick and Lehman 1999, Kessler et al. 2004, Bostock 2005. Salicylic acid is the corresponding elicitor of metabolites conferring resistance to pathogen attack. Notably, some evidence suggests a negative interaction between jasmonic acid and salicylic acid-mediated response epithways, with a corresponding trade-off in resistance traits such that resistance to pathogens confer susceptibility to herbivores and vice versa. Bostock 2005, Stout et al. 2006. Although there is evidence for positive crosstalk coordination among jasmonic acid, salicylic acid and other phytohormonal signals, e.g. ethylene, as effectors of gene expression for resistance traits, the functional link between herbivore and pathogen resistance is not always clear, but is an exciting and growing area of research. Kessler and Baldwin 2002, 2004, Bostock 2005. So far, we have discussed phytochemical signaling and induced resistance only in the context of within-plant processes. However, there is evidence that plants eavesdrop on herbivore-induced plant volatiles released from neighboring plants when they are attacked by herbivores. Baldwin et al. 2006, Kessler et al. 2006. The result of this priming is the activation of signaling pathways and ensuing defenses before herbivore attack. The hypothesis of talking trees was conceived decades ago, Baldwin and Schultz 1983, but is now gaining broader acceptance and may allow researchers to determine whether fluency, the ability to detect and respond to volatile signals from attacked neighbors, enhances plant fitness in natural communities. Engelberth et al. 2004, Baldwin et al. 2006, Kessler et al. 2006. Understanding the biochemical machinery and genetics underlying the expression of resistance traits to herbivores and plant pathogens should allow not only for the elucidation of constraints on the evolution of induced resistance, but also for its genetic manipulation. Kessler and Baldwin 2002, 2004, Taylor et al. 2004, Jalali et al. 2006. Breakthroughs in our knowledge of signaling pathways in plants have already led to important implications for agriculture. For instance, the use of natural elicitorsec as jasmonic acid has implications for management of pest herbivores. Toller 1999a, b. 2002a. Specifically, applications of jasmonic acid in tomatoes result in increased levels of proteinase inhibitors and polyphenol oxidases, metabolites implicated in resistance against insect herbivores. Importantly, jasmonic acid-induced plants can experience lower densities of herbivores and incur much less herbivora than control plants. Molecular techniques have been used to genetically engineer constitutive defensives into crop plants. Ely 1993, Gould 1998, Cerda and Pauletti 2004, Christou et al. 2006, Ferry et al. 2006. So-called transgenic crops contain resistance-expressing genes that have been artificially inserted into their genomes. The inserted gene sequence, the transgene, may derive from other organisms such as unrelated plants or bacteria, and organisms including transgenic crops containing transgenes are called genetically modified organisms. For example, corn, rice, cotton and many other crops have been genetically altered to express a bacterial endotoxin. Ely 1993, Christou et al. 2006, Ferry et al. 2006. Genes expressing the endotoxin are obtained from the soil-dwelling bacterium Bacillus thuringiensis, BT, a species that is toxic to many lepidopterans and is marketed worldwide as microbial insecticide. Gill et al. 1992, Entwistle et al. 1993. Millions of acres of crops engineered with BT endotoxin, e.g. BT corn, have been planted and have proved very effective against serious lepidopteran pests. Potential problems arising from BT crops include the killing of non-target organisms, the development of resistance in targeted pests and the migration of transgenes to other plants via hybridization and introgression. Tabashnik et al. 1991, Cerda and Pauletti 2004, Michaud 2005, O'Callaghan et al. 2005, Ando and Zwalin 2006. For instance, under artificial conditions, when genetically modified corn was first deployed, pollen expressing the Bt endotoxin drifted to nearby milkweed plants where it killed non-target herbivores such as monarch butterflies, Danaus plexippus, Lassi et al. 1999. Further risk assessment showed that the hazard was less than originally observed. Advances in Bt-engineered crops have resulted in plants that express the toxin only in certain tissues or crop stages targeted for pest consumption and not in pollen, Cerda and Pauletti 2004, Christou et al. 2006. Nonetheless, there is still concern over the use of transgenic crops, which has spurred much research on non-target herbivores and natural enemies as well as resistance management, Cerda and Pauletti 2004, Dively 2005, Ando and Zweilen 2006, Christou et al. 2006. 4.3.6 Herbivore counteradaptations to allochemical plant defenses. Insect herbivores have evolved a variety of biochemical and behavioral mechanisms that allow them to cope with the qualitative, quantitative and induced defenses of plants, Carbon and Agrawal 2002. These mechanisms include detoxification, excretion, sequestration, behavioral deactivation or avoidance, and host plant location. Detoxification is the most frequent mechanism used by both monophagous and polyphagous insect herbivores to overcome the qualitative defenses, toxins, in their host plants, Bratson 1992, Schuler 1996, Feyerisen 1999. The major detoxification enzyme systems in insects include cytochrome P450 monooxygenases, historically called mixed-function oxidases, glutathione S-transferases and esterases. P450 enzymes are a diverse class of enzymes that play a critical role in the degradation of allochemicals such as alkaloids, furanocoumarins and pyrethrins, as well as many synthetic insecticides, Barenbaum and Zengel 1994, Lee et al. 2003. Moreover, P450s can be induced after exposure to secondary metabolites and their induction is related to diet choice, Hung et al. 1997, Feyerisen 1999. For instance, Caterpillarsoft Manduca sexta increase their consumption of alkaloid-rich food only after enhancing thymidgut activity of P450s, and decrease their choice of alkaloid-containing food when P450 activity ice-blocked. Herbivores that specialize in toxic plants often employ other more specialized detoxification systems that allow them to metabolize defensive metabolites unique to those plants, Carbon and Agrawal 2002. A good example involves the bruchid weevil, Caredes brasilensis, which feeds very successfully on the Caredes brasilensis, which feeds very successfully on the seeds of the tropical legume, Diaclea megacarpa, seeds that are toxic to most herbivores because they contain high concentrations of the non-protein amino acid cannabinine, Rosenthal et al. 1982. Cannabinine derives its toxicity because, also an analog to the protein amino acid arginine, it becomes incorporated into polypeptide chains, thereby disrupting protein function. In C. brasilensis, RNA transferase discriminates between arginine and cannabinine such that cannabinine is not incorporated into polypeptides. Moreover, cannabinine is metabolized and the resulting nitrogen-rich compounds become sources for the synthesis of amino acids, thus resulting in great nutritional benefit. Insect herbivores have also evolved mechanisms to counter the effects of digestibility-reducing metabolites, quantitative defenses. Recall that tannins can reduce the digestibility of plant tissues by precipitating plant proteins. However, the precipitating effects of tannins are most effective under specific midgut conditions such as a low pH. Many lepidopterans offset the adverse effects of tannins by maintaining a high gut pH, surfactants or a specific redox potential, all of which can diminish the protein binding properties of tannins, Barenbaum 1980, Martin and Martin 1984, Apple and Maines 1995. By inhibiting specific digestive enzymes, proteinase inhibitors also interfere with the digestion of plant proteins. In response, several lepidopterans have apparently countered by developing alternative proteinases that are insensitive to inhibitors in their diet, thereby minimizing growth penalties, Broadway 1996. Metabolic counteradaptations to both activated and preformed induced defenses are also employed by insect herbivores. Regarding activated defenses, specialist herbivores exhibit feeding behaviors that apparently moderate the induction of certain allochemicals. For example, feeding by Manduca sexta larva on wild tobacco induces large increases in jasmonic acid pools, but there is little amplification of alkaloid production, Carbon and Baldwin 1997. Larvae apparently feed in a stealthy fashion, reducing the alkaloid response below that resulting from a comparable amount of artificial defoliation, Baldwin 1988. In this case, it isn't known how larvae interfere with the plant's signal transduction cascade. However, glucose oxidase, one of the principal components of caterpillar saliva, is responsible for suppressing induced resistance in tobacco by larvae of Helicoverpa z. Felton and Eichenseer 1999, Musser et al. 2002. This enzyme prevents the induction of alkaloids by directly inhibiting wound signaling elicitor jasmonic acid or by antagonizing its interaction with other signaling pathways. Specialist herbivores have defeated preformed inducible defenses such as the compartmentalized precursors, glucosinolate and myrosinase, to toxic thiocyanates in crucifers. For instance, larvae of the cabbage white butterfly, Pieris rapi, employ a gut protein that prevents the formation of toxic thiocyanates by redirecting glucosinolate hydrolysis to nitriles that are subsequently excreted, Whitstock et al. 2004. In contrast, larvae of Plutella xylostella, a lepidopteran also specialize to feed on glucosinolate-rich crucifers, desulfate glucosinolates metabolites that can no longer act as substrates for myrosinases, thus precluding the formation of toxic thiocyanates, Ratska et al. 2002. Likewise, larvae of the neotropical butterfly, Heliconius sera, employ a novel enzyme system that allows them to metabolize a compartmentalized cyanogenic glycosides present in their Passiflora auriculata host plant tothiols, Engler et al. 2000. Moreover, this process precludes therales of hydrogen cyanide HCN and alloscaterpillars to use the otherwise toxic compound ACE nitrogen source. Other herbivores employ excretion as a mechanism to cope with secondary metabolites. For instance, larvae of the tomato hornworm, Manduca sexta, have evolved an excretion system that eliminates non-metabolized nicotine much faster than other herbivores due to a specialized alkaloid transport system in their Malpighian tubules. Malpighian tubules are the excretory organs of insects that remove nitrogenous waste products, also toxic alkaloids, from the hemolymph and deposit them in the hindgut, where they are eliminated. Many phytophagous insects, especially monophagous species, sequester plant-derived metabolites either as a non-toxic form in the hemolymph or as toxins stored in the cuticle or in specialized glands or organs where their potential toxic effects are isolated. Bloom et al. 1990, Bowers 1992, 1993, Dyer 1995, Carbon and Agrawal 2002. Sequestered toxins include the cardenolides deposited in the integument of monarch butterflies, Dinosplexipus, and milkweed bugs, Onchopeltis fasciatus, iridoid glycosides present in the integumentoph checkerspot butterflies, Euphydryas phaeton, glucosinolates stored in the hemolymph of sawflies, Athalia rosae, pyrrolizidine alkaloids in the hemolymph of leaf beetles, Orina cachalii, and phenolic glycosides present in the eversible glandsoph larval chrysomelid beetles, Photora vitellinae, Brower 1984. Bowers 1992, 1993, Pastiles et al. 1983, Hartman et al. 1999, Trigo 2000, Mu diaresis Yair et al. 2001. Although it is tantalizing to view sequestration as a metabolic process by which herbivores avoid plant toxins, most evidence suggests that the primary selective force underlying sequestration is defense against natural enemies. Pastiles and Gregoire 1983, Dino et al. 1990, Carbon and Agrawal 2002. For example, many herbivores that sequester toxins are warningly colored, aposematic, and unpalatable to predators. Moreover, although many sequestered toxins are acquired during the larval stage, some adult butterflies Daninae and Ithamyainae and moths, the Arctiid Eudothesia ornitrix, derive thermostats from toxins e.g., pyrrolizidine alkaloids ingested in the nectar of their floral resources on not from larval host plants, further implicating predation in the evolution of sequestration. Bowers 1993. Moreover, sequestration is a costly process and its advantage may be realized only in the presence of natural enemies. Carbon and Agrawal 2002. Nonetheless, until shown otherwise, sequestration is a possible mechanism by which plant toxins are rendered innocuous. Insect herbivores have evolved a suite of behaviors, so-called counterploys, that allow them to avoid or diminish the adverse consequences of secondary metabolites, particularly quantitative defenses like resins and latexes that are contained in ducts or canals. Dussord 1993, Payne et al. 1997, Carbon and Agrawal 2002. A classic case involves bark beetles Coleoptera, Scelididae that employ an aggregation pheromone to mass attack single pine trees. Seibold et al. 2000, Keeling and Bowman 2006. The synchronized attack by thousands of individuals burrowing into a single tree depressurizes the resin canal system, diminishes the pitching out of oleoresins that potentially might entrap beetles and facilitates tree colonization. Notably, in some bark beetle species, aggregation pheromone, which is produced by first arriving colonists, is metabolized from terpenes in the tree under attack. Other mandibulate herbivores deactivate canal-borne defenses in many latex or resin-bearing plants by clipping veins or trenching entire leaves before they feed distal to the cuts on leaves. Dussord and Eisner 1987, Becerra and Venable 1990, Becerra 1994, 2003, Dussord and Dino 1991, 1994, Dussord 1993, Figure 4.10. Vein cutting and trenching block the flow of latex or resin, rendering otherwise defended leaf tissues suitable for feeding. Vein cutting behavior occurs commonly in herbivores associated with milkweed plants, Apocynaceae, such as chrysomelid beetles, Labidomera clivicalis, cerambycid beetles, Tetraopes tetrophthalmus, and Danade butterflies, Danaus gillipus and Diplexipus, but has been observed in numerous other families of latex or resin-bearing plants, e.g., Anacardiaceae, Apocynaceae, Burseraceae, Moraceae. In contrast, trenching behavior occurs on a mutually exclusive group of plants, e.g., Apiaceae, Astraceae, Caricaceae and Cucurbitaceae. Notably, there is amazing correspondence between behavior and the architecture of the canal systems in latex-bearing plants. Dussord and Dino 1991. Vein cutting occurs in plants with branching canal architecture, whereas trenching is employed by herbivores on plants with net-like canal systems. Generalist caterpillars which lack these behavioral counterploys perform very poorly on intact plants, but many of them can feed successfully if latex-bearing plants are experimentally depressurized. Dussord and Dino 1994. The evolution of vascular sap feeding has allowed these herbivores to largely avoid chemicals that are compartmentalized in non-vascular tissues. Raven 1983. For instance, by feeding in the phloem of tobacco, green peach aphids Mysis persicae avoid ingesting nicotine that occurs in other plant tissues. Guthrie et al. 1962. Last, adapted specialist herbivores have turned the tables on plants by cueing in on volatile plant toxins to locate their specific host plants. Visser 1988, Bernays and Chapman 1994, Renwick 2002, Bruce et al. 2005. Host selection is a complex continuum between two extremes, namely herbivores choosing their host plant from a distance using olfactory and visual cues, and herbivores selecting their host after contact using gustatory information. The allochemicals in crucifers Brassicaceae are involved in both processes. For instance, glucosinolates often serve as oviposition or feeding stimulants, whereas their volatile hydrolysis products isothiocyanates are attractants for several specialists, such as cabbage aphids Brevicorine brassicae, flea beetles Philotreta spp, Diamondback Plutella xylostella, and cabbage. Figure 4.10 Deactivating the defenses of latex-bearing plants. Insect herbivores nip the main midrib of their host plant with their mandibles, cut off flow of latex beyond the cut and subsequently feed distally where they avoid the latex. Caterpillars of Danauscylopus on Asclepias carassivica, Dussord and Eisner 1987 b. The milkweed beetle. Butterflies Pieris rapi, Renwick 2002, Bruce et al. 2005. Similarly, bark beetles e.g., Ipes spp. And Dendroctonusp. Use conifer-produced terpenoids to facilitate host selection, colonize particular tree species and identify weakened trees that they can more easily colonize. Payne et al. 1997, Seibold et al. 2000, Raffa et al. 2005, Keeling and Bowman 2006. Several bark beetles of Venus metabolize conifer-derived terpenoids as attractants and aggregation pheromones to facilitate mass colonization of trees. It is important to realize, however, that host plant recognition often depends in ratios of plant volatiles and not just in the detection of particular compounds. 4.3.7 Leaf abscission is a plant adaptation to deter herbivora. Premature leaf abscission is thought to be an adaptive response to herbivore attack, which has particularly adverse effects on immobile herbivores such as gall inducers and leaf miners. Feith et al. 1981, Stilling and Simberloff 1989, Connor and Taberner 1997. The gall-inducing aphid Pemphigus batae, for example, induces premature leaf drop in cottonwood trees populace, reducing aphid populations by more than 50%, Williams and Witham 1986. The induced response is density-dependent. Leaves with 3 or more galls are 4 times as likely to be dropped as leaves with 1 gall, and 20 times as likely to be abscised as ungalled leaves. Remarkably, 98% of the gall aphids that dropped to the ground died. For leaf-mining herbivores, early abscission can also result in reduced body size, lower fecundity, and increased exposure to predators, although the latter is not always the case. Waddell et al. 2001. Not surprisingly, plant growth hormones such as cytokinins, hormones that regulate leaf abscission, do occur in the ovipositional secretions of gall-inducing sawflies. Horry 1992, Schultz 2002. Thus, the biochemical machinery necessary to direct leaf abscission appears to occur in herbivores that are particularly vulnerable to leaf drop. 4.3.8 Encouraging predators, parasitoids and mutualists as a defense against herbivores. Plants benefit from their association with enemies of insect herbivores, an important element in reducing herbivore damage. However, this large topic will be discussed more appropriately in Chapter 6 on mutualistic associations, and in Chapter 13 on multitrophic level interactions. 4.3.9 Spatial and temporal variation in plant allochemistry and nutrition pose tracking problems for insect herbivores. We have just explored the tremendous variation in the types of defensive compounds and nutrients that occur in plants. Despite the counteradaptations that insect herbivores have evolved to cope with this chemical diversity, they still face tremendous spatial and temporal variation in plant nutrition, allochemistry and morphology, making it. Caption for Figure 4.10 Kant. Tetraopes tetrophthalmus, on Asclepias syriaca, Dussord 1999, C. The katydid, Scuderia furcata, on Apocynum cannabinum, Dussord 2009, and D. The leaf beetle, Labidomera clivicalis, on Asclepias syriaca leaf, Dussord 1999. E. By trenching leaves of Lobelia cardinalis, the Lepidopteran, Enigmagramma basigera, cuts the flow of latex to leaf tissue within the circumscribed trench where it then feeds. Dussord 2005. Larvae of Trichopluginae expend an extensive time and energy trenching leaves of prickly lettuce, Lactuca seriola, before feeding distal to the trench. Shown are larval trenching efforts after 3 passes F and 15 passes G. Dussord and Dino 1994. A. From Dussord, D. E. and T. Eisner. 1987. Vein cutting behavior. Insect counterploy to the latex defense of plants. Science 237, 898-901. Reprinted with permission from AAAS. C. Reprinted with permission from Blackwell Publishing. All photographs are Dr. David Dussord. See color plate section. Fundamentally difficult for them to locate optimal resources for oviposition and development. Dino and McClure 1983a, b. Failure to do so can have very negative effects on development and survival with extended effects on population dynamics. Variation in plant defenses and nutrition can be genetic or environmental and can occur within a single plant, among plants in a population, and across plant populations at larger habitat and geographic scales. Dino and McClure 1983a, Fritz and Sims 1992, Hunter et al. 1992, Orions et al. 2003, Johnson and Agrawal 2005. In the following sections, we elaborate once sources and spatial scale of such variation and discuss its consequences for herbivores in the context of offsetting the apparent advantage insects have over plants with their short generation times and high recombination rates. As individual plants develop, there is remarkable spatial and temporal variation in tissue chemistry, morphology and phenology with which herbivores must contend. Hunter et al. 1997, Carbon and Toller 1999, Brennan et al. 2001, Lawrence et al. 2003. In a spatial context, young terminal leaves are often more nutritious and less tough than mature leaves. Raup and Dino 1983, Amack and Leather 2002, Peters 2002. Based on these two plant characteristics, herbivores should benefit by feeding on young leaves. However, certain allochemicals that inhibit herbivora growth, such as flavonoid and terpenoid-based resins, are often more concentrated in the terminal leaves of plants. The elevated resin level denies herbivores access to nitrogen-rich and tender terminals and forces them to feed on suboptimal basal leaves. Larvae of both diprionid sawflies, Neodyprion swaini, on jack pine, Pinus banksiana, and the checkerspot butterfly, Euphydryas chalcedona, once chaparral shrub, Diplacus aurantiacus, face this feeding compromise. Akeda et al. 1977a, b. Lincoln et al. 1982. Likewise, in apple trees, the phenolic compound phloretin is more concentrated in terminal leaves and the within-tree distribution of phenolics is associated with the occurrence of apple-feeding herbivores. Hunter and Hull 1993, Hunter et al. 1994. Overall, spatial variation in secondary metabolites is thought to be an important factor influencing the distribution, abundance and population dynamics of insect herbivores. Hunter et al. 1996, Forkner et al. 2004. Moreover, there is remarkable ontogenetic and temporal variation in leaf chemistry and morphology that places further constraints on insect herbivores. Dino and McClure 1983b, Krishik and Dino 1983, Forkner et al. 2004. A striking case of ontogenetic variation that leads to dramatic structural differences and several chemical discrepancies, e.g., tannin content between young and mature foliage is called heteroblasty. Brennan et al. 2001, Lawrence et al. 2003, Grau et al. 2005. The genus Eucalyptus contains many species that exhibit this phenomenon whereby foliage changes rapidly from juvenile to adult leaves as trees age. This ontogenetic shift has dramatic consequences for herbivores. For instance, the psyllid Tenaritana eucalypti develops only on juvenile shoots of E. globalus, whereas its congener C. spatulata is restricted to adult foliage. Similarly, one of the commonest herbivores in the same heteroblastic eucalypt is the leaf beetle Chrysophtharta agricola, which is 9 times more abundant in the adult foliage zone of trees as it is in the juvenile zone. Thus, as trees undergo heteroblasty, there are dramatic shifts in the composition of the herbivore assemblage. There is also remarkable within-plant variation in plant chemistry and morphology at the seasonal scale. As the season progresses, leaves generally become less nutritious, tougher and more concentrated in digestibility-reducing metabolites such as tannins and resins, although there are certainly exceptions and much variation among specific defensive compounds. Krishik and Dino 1983, Raup and Dino 1983, Amack and Leather 2002, Salmonen et al. 2004. Varley, 1967, was the first to observe that the common herbivores such as wintermouth, Operaphthora brumata, on English oak Quercus feed early in the season just following bud break, whereas many rarer lepidopterans feed during the autumn. Feeney, 1970, explored this pattern by measuring a variety of plant parameters and concluded that many common herbivoraceae have adjusted their life histories to feed in spring when leaves are most nutritious and tender, and when tannin concentrations are lowest. More recently, seasonal increases in condensed tannin content have been reported for several North American oak species. Forkner et al. 2004. Moreover, feed densities of several of the abundant herbivores want these oaks are negatively correlated with the tannin concentration. In a similar example, overwintering caterpillars off the checkerspot butterfly, Euphydryas chalcedona, emerge from diapause in spring to feed on the nutritious foliage of Diplacus aurantiacus when the concentration of leaf resin ice at its seasonal low. Mooney et al. 1980, 1981. The important take-home message is that many insect herbivores own oaks and other plants face a very narrow seasonal opportunity for development on optimal resources. They are precluded from feeding very early in the season by bud scales that deny access to forming leaves, and after bud break they face the rapid onslaught of increasing mechanical and constitutive defenses as the season progresses. Failure to time oviposition and development with optimal leaf resources during the narrow spring window can have very negative consequences for growth and survival. Hunter et al. 1997. Immobile herbivores such as leaf miners and gall inducers can face further constraints at the end of the growing season when deciduous trees drop their leaves, thus cutting the growing season short and often inflicting high levels of mortality. Feith et al. 1981, Williams and Witham 1986, Waddell et al. 2001. An example involving the gall-inducing aphid Pemphigus vitae on narrowleaf cottonwood, Populus angustifolia, highlights the difficulty that herbivores have in tracking suitable resources in a spatial and temporal mosaic of changing leaf morphology and chemistry. Witham 1978, 1979, 1980, 1983, Zucker 1982. In early spring, when buds just begin to break, female aphids, stem mothers, hatch from overwintering eggs on the trunk, migrate to developing leaves and begin to initiate galls. Females methodically probe developing leaves with their stylets, and the proliferating gall tissue soon encloses the aphid. She then produces up to 300 progeny parthenogenetically that develop within the gall. All colonizing females have settled and begun gall initiation within a 5-day period in early spring, and multiple females can initiate galls on the same leaf. The success of gall formation and progeny development, however, is very dependent on leaf size and gall position on the leaf, both of which vary in the concentration of phenolic glycosides. The basal position of the largest leaves on the tree is the optimal site for gall induction, and colonizing females engage in competitive shoving matches to win access to such sites for gall initiation. Figure 4.11a. On the largest available leaves greater than 15 square centimeters, the probability of females dying during gall formation was 0%, whereas 80% of colonizing females die colonizing small leaves, less than 5 square centimeters. Similarly, female body weight and progeny number were 70% and 220% greater, respectively, on large leaves than on small ones. Remarkably, only 1.6% of all available leaves fall into the most suitable size class, as most leaves are much smaller. Within a leaf, females inducing galls at the leaf base were larger, died less frequently and produced more progeny than females initiating galls in more distal positions, figure 4.11b. Not coincidentally, the concentration of phenolic glycosides is lowest where aphid performance is best, namely on the largest leaves and at the base of individual leaves. 4.3.10 Sources and consequences of variation in plant resistance, genes and environment. Because secondary metabolites and nutrients often determine the resistance or susceptibility of plants.